Hi, my name is Isabelle DeMarco, and I'm a second-year biochemistry major working in the Chemi lab. Today I'm going to be talking about my research in developing an assay for break-induced replication in Arabidopsis. Our DNA is constantly under attack. Exposure to radiation, chemical agents, and even regular cellular processes can cause breaks that our cells must repair. One type of break is a double-stranded break, where both sides of the DNA double helix are damaged. When this happens, it can be repaired in one of two ways. The first is called non-homologous end joining, where the broken ends of the DNA are brought back together and repaired to be as they were before. The other is called homologous recombination, in which the broken DNA finds a complementary sequence on the homologous chromosome. It invades the homologous DNA and uses it as a template for repair. One particular type of homologous recombination is called break-induced replication, or BIR, and this can occur when a homologous template is only available on one side of the break. The broken DNA strand invades the unbroken strand. However, the repaired strand will not rejoin with the broken strand, and the DNA is replicated all the way down the chromosome, resulting in the repaired DNA segment being identical to that of the homologous chromosome. One issue with BIR is that it is not very precise. It has been observed in yeast that BIR causes many mutations which appear similar to mutant patterns seen in human cancers. Since BIR does not occur very frequently, we don't know a lot about the mechanisms underlying it or why it results in so many mutations. In order to understand it better, we need a way to study it. Recently, the Chemi lab characterized the spontaneous correction of a chromosome translocation in potato through a mechanism that resembles BIR. We think that this is the first documented example of BIR in any plant. To study how it may work, we need to make it happen again. Now we are working on an assay to induce BIR in Arabidopsis using CRISPR. By integrating the Cas9 gene and a gRNA sequence into the Arabidopsis genome, we can direct the Cas9 protein to cut the DNA at a specific location on a chromosome. Various repair techniques may be used to fix these breaks, but we want to focus on BIR. To know that BIR has occurred, we will track a gene that controls the shape of Arabidopsis leaf hairs, which are called trichomes. Loss of this gene by BIR changes the trichomes from a three-pronged hair into a spike. This will allow us to identify BIR simply by looking at the plants. Having a mechanism to induce break-induced replication at high rates will enable further study into this largely uncharacterized genetic phenomenon. Thanks for watching, and a special thanks to everyone who has helped make this project possible.